Good morning, everybody. Thank you very much for joining us for this webinar today. It's 9 a.m. here in San Diego, and today's webinar is on the subject of ICM 3D ligand editor, where we'll be covering ligand docking, editing, and optimization. This is a first in a series of 6 webinars where we'll be focusing primarily on ligand docking and virtual screening. So today's webinar is here, and in 2 weeks' time, every 2 weeks, we're going to build up from docking to preparing chemical libraries for virtual screening, millions of compounds, then on to more technical stuff such as receptor flexibility during ligand docking and screening. And then we will finish the series showing ultra-large library screening using GPU methods and AI methods in ICM. So if you have any questions during the webinar, please use the Q&A option in the Zoom. I don't think the chat is working. It's disabled, but the Q&A should work. Or you can ask questions after the webinar, info@molsoft.com. A recording of this webinar will be available on our YouTube channel. You can just search Molsoft and you'll find it. And we also provide a free 2-week trial license of the software, which you can start at any point during the series. Just get in contact with us at info and let us know you would like info@molsoft.com and let us know that you would like a license key. And the software runs on Windows, Mac, and Linux. So in this webinar, we will be covering the ligand editor. So the ligand editor is an environment for you to explore in 3D different modifications to lead compounds. There are a variety of, so I'll show you how to set up the ligand and the receptor for docking and minimization. But once you have it set up, you can display different pocket surfaces. You can display hydrogen bonds, other interactions, which we'll go through. We'll also show how to make modifications in 3D or in a 2D environment, and then see the effect of those modifications on ligand binding. And then show how you can save and share and export your results. If you're not familiar with Molsoft, we were founded by Ruben Abagan back in 1994, and there's some key papers which describe ICM. ICM stands for Internal Coordinate Mechanics, which we use rather than Cartesian coordinates. The main scientists involved in the development are Ruben Abagan, who's our founder, Max Totroff, who's our CTO, and Eugene Rausch, who is our principal developer, and Polo Lam, who's a senior scientist here at Molsoft. So over the years, we've competed in docking challenges, and ICM did very well in the very early ligand docking challenges, and so ICM was used by some big pharma initially, and that's how it got its name. And over the years, we've had long-term collaborations with big pharma such as Novartis. For example, we developed Focus, which actually contains the 3D ligand editor, which we're going to look at today. And then more recently, we've been working on AI and deep learning methods for ligand docking and modeling. And we also had a very good success stories in the industry-wide D3R docking challenges that were a few years ago now, but they're still relevant. But and a variety of different drug design projects and published stories that we can show you. I can send papers for. So the ICM Pro desktop modeling software We're today just focusing on the ligand editor, but there's a whole variety of tools from molecular visualization and from and for X-ray structure analysis, so you can read in maps and build biomolecules. You can analyze the structure, identify ligand binding pockets and protein-protein interaction sites. There are tools there for homology modeling, loop modeling, building a full atom representation of your protein. There's molecular dynamics, Monte Carlo. So these are all tools we probably won't cover today, but they're available if you have the trial license. There's bioinformatics tools, so you can build alignments and propagate those alignments to the structure. So you can identify where there's maybe similarity between multiple crystal structures and around the pocket, for example, shown here. There's also a whole suite of cheminformatics tools where you can calculate properties, generate multi-parameter optimization scores, generate combinatorial libraries, analyze SAR, and build 2D and 3D QSAR models. And then there's tools for docking and virtual screening. So you can dock small molecules, you can dock peptides, fragments, and protein-protein docking. So the 3D ligand editor is described in this paper. This was a collaboration, long-term collaboration we have with Novartis, which this paper describes the development of this environment, as well as some of the cheminformatics tools that we have. So the 3D ligand editor is a fully interactive tool. So it basically allows you to easily sort of generate ideas. So you can read in a co-crystal structure, or you can read in an apostructure and dock a ligand. If you read in the co-crystal, you can make modifications. You can change atom types, bond types. You can screen groups at particular points. You can also do a core replacement and screen a database of different cores. And the advantage of this method is that you immediately see the effect of the modification on predicted binding, on the binding score. And there's also full undo and redo capabilities, and the modification history is fully saved. Just wanted to point out that you can also do what we're showing today. You can also do on a web page using the JavaScript version of ICM. But we'll be showing it inside ICM, the main desktop modeling software. So the example we're going to look at today is some EGFR inhibitors with quinazoline derivative core, these are the first generation inhibitors for EGFR that were designed for these common mutations such as exon 19 deletion and exon 21 insertion, which drives about 90% of non-small cell lung cancer cases. These first generation EGFR kinase inhibitors are designed to inhibit EGFR by competing with ATP at the kinase catalytic site. We'll see later on that one of the main issues with these is the development of resistance mutations, such as the gatekeeper residue. And after about a year of use of the drug, sometimes these mutations occur, and we'll look at reason why. So, first step, if we're going to basically read in alotinib, which is a first generation EGFR inhibitor. and we're going to make some modifications to that inhibitor inside ICM. So we open up ICM. Hopefully you see the ICM screen here. And we have up here a PDB search option, and there's a search tab here, PDB search. So we could effectively just type a string like EGFR here and then search, and then we get all the EGFR structures that are in the protein data bank. We can sort them by resolution if we wanted to. Here, there's also a column with the ligands that are bound to that receptor here. Or alternatively, we can click on this search button with the ring and the pencil, and that will open up a window like this. And we could search for allotinib, which is the ligand we're going to work on today. So you go to the chemical search button here, which has a dictionary of structures, and it will draw the allotinib structure. You can see on the right-hand side, there's some properties that are automatically calculated for this structure. We just go close and say changes yes. And you can see now in the search tab here, we have the SMILE string for that inhibitor. We just go here. And we get a new table with the search results. There are 3 structures in the PDB with this ligand bound. And the one we're going to use is 1M17, which is the higher resolution structure. We can load that structure by just double clicking here, or probably the easiest thing, which maybe we should have started with the beginning, was just type the code in. So 1M17, you can type the code in here in the PDB search here and press the binoculars button. And this loads the structure. So you can see that we have the one protein chain, which is shown in blue. We have the ligand here. If you can just expand it and you can see the ligand. And then we have the water molecules that are provided with the crystal structure here. So we're first going to set up, we're going to edit this inhibitor. So we go to the, you'll see at the top here, we have a tab called Lig Edit here. So we single click on that, and that opens up these tools shown at the top here on this bar, and it gives you the environments. This is the environment we're going to work in inside ICM when we're using the 3D ligand editor. Later in the series, we'll be using the docking menu. This gives additional tools for virtual screening and other docking procedures. The ligand editor is more designed for small scale docking and optimization and interactive modeling. So our first step is to tell ICM which we have these 2 buttons at the beginning. These are the buttons that help us set up the ligand and the receptor. So we first click on the ligand button here, and it opens this this window, and it asks us for the ligand molecule. There's only one ligand molecule in this protein structure, so it finds it automatically here. And inbuilt in ICM, we have a pKa model, and so it will charge your ligand based on that pKa model. But if you've prepared this in another software, you can uncheck that option. And we just go OK. Now you can see in the ICM workspace on the left-hand side, we have our original crystal structure. And now we have another object called 1M17_lig. So this is the ligand that we're going to edit. It just keeps the crystal structure as a reference. Now we need to tell ICM which crystal structure we want to use for the modeling that we're going to do. And that's the second button along. We just click on that one and this one here, and it brings up this panel. It asks us which is the receptor object. Again, there's only one loaded in ICM at the moment, so it finds it automatically. If you have more, you can use the drop-down button and select it. Then we have some questions about the waters. Generally, we would recommend to keep the tight waters, those that are making 3 to 4 hydrogen bonds, but you can say you may have already carefully looked at the pocket and decided which waters you want to keep and deleted the others, which is an option from the PDB file, and you can say keep all waters, or you can just assume that when you dock that the waters are going to be displaced, and so you're going to delete all the waters. It's kind of a subjective thing, and we'll talk a little bit more about that in the next webinar. We're adding, because we're going from a crystal structure, we're adding hydrogens, so we need to optimize the hydrogens. There are also some ambiguities from the crystal structure, such as the orientations of asparagines and glutamine residues. This option mark missing side chains. Basically, if there's 0 occupancy on some atoms on a side chain, then you won't see if you have this checked, you won't see an atom there. It'll be masked out. If you uncheck it, ICM will build the full atom representation of that residue for you. But hopefully you're modeling to a relatively high resolution structure. So in most cases, the occupancy of the side chains is pretty decent in the binding pocket, but that's something to be careful about. We have a function here called identify binding pocket box, which uses the ICM pocket finder method, which will identify allosteric sites and orthosteric sites and hidden cryptic sites if you've got MD or models. But in this case, we know where the binding pocket is because we have the ligand bound. So we can just choose the option shown here called make box around existing ligand. So just single click here. Then you see in the graphics that we have this purple box. So in this purple box, we're going to build grid potential maps for the docking. So we have things like maps for van der Waals interactions, so the attractive and repulsive forces between the atoms. There's a map for electrostatic interactions, and hydrogen bonding contributions from donor-acceptor pairs. There's desolvation maps, so the energetic cost of removing water molecules from the binding site. And when we dock, we also have a penalty for flexibility, so the loss of degrees of freedom of the ligand upon binding. So this box is kind of important, especially if you're designing, if you're going to try and make your ligand larger. So you need to make sure that the box kind of encompasses the region you want to target. So you can see here we're looking at EGFR, and we have the N terminal here. We have this alpha C shown here, and the C terminal here. We have the P loop, glycine-rich loop, dropping down here. So we may want to, if we want to extend our molecule further towards the alpha C, we may want to make this box a little bit larger. So you can drag on the corners to make the box a little bit larger. And it gives more space. So when you're happy with that, you just go OK, and it creates the maps. So this is the initial setup in the ligand editor. We have in the ICM workspace, we have the ligand, which we're going to model. The receptor, we're going to model the ligand in. Our original PDB file. And just for reference, we have those maps which I described, but you don't need to see them. It also, by default, generates this pocket representation, which is a very useful pocket representation to have when designing new leads. So we'll talk about a little bit about it in a minute when we get onto that. So we have for the ligand receptor interaction tools, you can display the ligand surface, which I showed, the receptor pocket. So these are colored by binding property. So green is hydrophobic, blue is hydrogen bond acceptor, donor, and red is hydrogen bond acceptor, and white is lipophilic regions. So you can highlight the pockets, you can display hydrogen bonds, you can show energy circles, which will show any strain or clashes with the receptor. You can compare your docked ligand with the relaxed conformation of the ligand, which is sometimes useful when generating new leads. You can highlight unsatisfied hydrogen bonds, which may give you some clues where you could build onto. So all the display options inside the ligand editor are in this button here with the drop down here. So there's an option called Pretty View, which I'll choose. I'll just click on it. It generates this representation. I don't like the spheres, so I can the CPK, so I can just go to Display tab and choose and uncheck the CPK option here. Yeah, so we have the ligand shown here. So we can clearly see that this this inhibitor makes this is the adenine binding pocket of the ATP site. So this is the hinge, and so this quinazoline ring group is making a interaction with the hinge here. We have the glycine-rich loop, and it's going to make the display a little bit different to add a bit of transparency to the. So make the ribbon transparent. Make the ribbon transparent, and just change the depth for the transparent. Okay, so we can clearly see our ligand that we're going to change. So good practice when you after you set up the ligand editor is to just do a small minimization of the ligand, because we've added hydrogens to the protein structure. So we just want to make sure we sort of optimize the best pose for the ligand, and maybe iron out some little strain issues. So we can just minimize the ligand. There's two buttons here. One is for minimization and one is for docking. So if we click on this one, which is the minimization one, that will do a small minimization. And then we see at the top here, we have the ICM score, which we'll keep an eye on when we start modification. Some other visualizations we can do is we can visualize the receptor pocket shown here. So this is colored by the properties of the receptor. So we have hydrogen bond acceptor here. Blue is hydrogen bond donor. And we also have the option for the ligand pocket surface, which I'll just check. We already have shown here. So this is a great view. So it gives you an idea of where there's cavities to build. And you can see that there's space here out into the solvent accessible area here. Around this ring, there's a little bit of space, and here as well, there's some cavity you could build into if you wanted to. If we go to the back of the pocket, this shows that, you know, if you're adding a hydrogen to this, adding anything to this hydrogen, you're likely to clash with the receptor because there's really no space to build into. So other visualization tools include displaying the relaxed ligand, which I'll show you later. You can also analyze the contacts. So if we click on Analyze Contacts, it will give the sticks a zoom level depending on the amount of contact they make with the ligand. So go OK, and we get this table here. And so in the table, we can see that for each residue, we have the contact area, the exposed area, and percent buried, and the distance from the ligand. And in the graphics, I'm just going to undisplay the. So the residues that are making the biggest contribution to the contacts. So, for example, this residue here, it's displayed thicker. So this is a leucine, and so thinner ones are making weaker contacts. So it's just a useful way of seeing the contacts. You can also build a 2D representation. If we click on this button here, it will generate a 2D interaction map between your ligand and the receptor. You just go OK for the default values, and you'll get an image like this. I just need to move the screen up so it has if there's pi-cation interactions, hydrogen bonding, hydrophobic shown here. And this can be saved as an image if needed. Okay, so now we're all set up really. You can just a final thing for the visualization tools is that a hydrogen bond, you can toggle the hydrogen bonds on and off here using this button here. So now we're ready to make modifications. So that you'll see this panel in a minute, and this panel will give you the options for making modifications. So you can change atom types, you can change the charge, you can rotate torsion angles, you can change bond types, switch stereo, cis and trans change. And then we have this small a selection of substituents that you can dock on the fly. You can add new substituents to this, or you can screen our larger database of more than 64,000 substituents on the fly on your structure, but we'll show that in a bit. You can also make modifications to the ligand in 2D using our 2D editor, and then convert that to 3D. So if you make the edits, and then we click here, and it will dock that ligand and give you the score. You can also just completely delete the ligand and add a completely new ligand using the dictionary. Okay, so we now are ready to make modifications. So to make modifications, we need to open the modification panel, which is this button with the pencil. So you single click, and it opens this panel. So we can, most simple use of it is you can take your, so you can just to be sure we can see at the top here, we have the score, RTCNN score, which we'll discuss next week, 2 weeks time, which is our neural network score. But basically this is derived based on the protein data bank, and it's learned the interactions, and it's a good score in combination with our doc score, which is our physics-based score. So basically, the lower these scores, the better. We also report the ligand strain here as well. So if it gets beyond maybe 6 or 7, then you may have a very strained ligand, but we'll see ways of combating that. So simple way of using it, you can just take the atom, sorry, the atom panel here, just choose oxygen, for example, and you can change an atom to oxygen. So we can change this hydrogen just by clicking on it to an oxygen. We can evaluate the score of that change. I think it made it worse, so we can undo it. We can choose a different hydrogen. Try it here. And then you can do a small minimization using the button here. You can see with that modification, we added a hydrogen bond with this this group, part of the DFG region. So at the moment, we're looking at this, the orientation of this in terms of kinases. You have your adenine binding site here. You have the solvent exposed region here, where usually the ATP phosphates are and the sugars. And then at the back, we have the hydrophobic site, and there's an additional hydrophobic site here, which we can see. So we're making modifications in this region to start with. So that's just a simple atom. If you wanted to maybe change, we got an amine linker here. It's quite good because it gives a bit of rigidness to this molecule, but you could change it to like a thioether or something if you just click on the sulfur here, and then you would minimize and evaluate the score, or you could change it to an ether linker here. And if you don't like the changes, you just undo, go back through them, go back to the original one. I'll remove that oxygen as well, so undo. Yeah. So we showed that pocket surface. So if we did try to add a group here. So to add a group, you just click on the group you want to try and add. And if we try and fit it on here, it's going to try and dock it, and it will show us that that's not a very good modification because you have clashed with the receptor. It's shown by this. So we can undo that clash. And that was reflected in the score as well when you add that group. So we may want to try some modifications at this alkyne region on the meta position of this ring. So we can do that. If we can use the, if you wanted to, we can use the eraser button shown here, and we can delete atoms. So we can delete the alkyne group just by click on the eraser and then click on the atom. So we have this hydrogen. So we could explore some modifications at this point here. So one thing you could just add manually some different atoms, or you can choose from this panel. So we can select some halogens here. You just single click. You can choose a sulfonamide, sulfonyl, these groups here, or any number you want to try. And then you go back to the hydrogen here and just single click, and it will dock those modifications to that site. When it's done, you have this table shown here. It's ranked by score. So the lower the score, the better. So we can see that in this table, if we scroll down, that the bromine, chlorine modification are all better than the original hydrogen. So it all improves the score. But when we add a sulfonamide group, the scores come higher. So probably not as good modification. So if you wanted to load the bromine, we just double click and it loads that into your structure. If you wanted to have a look just to check the sulfonamide and load it, and you can see that the sulfonamide is making a clash here with the receptor. And in one of the future webinars, we'll be covering flexible receptor docking. So it allows some induced fit in the binding pocket, or you can dock to multiple conformations of your receptor. But here we're just doing it with one receptor at the moment. The ligand is fully flexible. So if we like the bromine modification, we can save this modification to a chemical spreadsheet so we can share it with our colleagues. So to save this modification, we have this table, obviously, but we can just, there's a button here that, zoom in, this button will add it to a table called Ligand. So this is just a record of any modifications that you actually like and you may want to go back to. They'll be stored in this table. You can also tag, you can tag them to give them a label. So you can say meta position changes or something like that. And that will add that tag here. So you can, if you have many hundreds of them, you can easily sort them by the modification that you're doing. Okay, so we've added this bromine. I'm just going to undo. We can undo this modification, go back to our original ligand. You can also click and hold on the undo button, and you can see there's an option called modification history. So this will give you a history of everything you've done and the table. So we had, the first thing we did was minimizing the grid. We erased some atoms. We attached a chemical group. And so that's all saved here. So you can just double click and it will load, go back to that position or that compound if you want. But we know that these this position seems to like halogens, right? So maybe some lipophilic molecule there might be a good option. So we do have this table. I'm just going to check. So we have these built-in substituents that you can use, or you can add your own one here. If you click on add new, you can sketch a modification that you want to add like this, and make an attachment point. So you right click and choose attachment point, and then you can just add it to the table here. So we've gone, but is here. But if you want to search a larger, extensive database of substituents, you can do that using the you can go to the Lig Edit, go to the Advanced button here and choose Find Group. So I'm going to go Find Group. And we have this table. It has 60,000 substituents. I was going to try some small lipophilic groups at this point. You can see when I click on that filter, it shows that there's 283 groups in this that are classified as small lipophilic, and we can screen them to that position. So we just select the atom we want to screen to. So there's a pick atom button here, which will just give you that green cross on the atom. So you go to the Advanced, Find Group, and we're going to do this screen on the fly. It should take 30 seconds, 40 seconds, we'll see how it does in the live. But it's running at the moment. You'll see some progress bar here. While it's running, we can just look at this Find Group. There's also options to filter by small donors or small acceptors, negative charge groups. Or you can just not filter at all and just screen all 60,000. It will take about an hour or so to do. So we screen those, and it's finished in under a minute. And we get this table called modifiers tab. It's going to delete these other ones. There's too many. So you can see we have a column with the RT-CNN score, which is a neural network score. So the lower the score, the better. We have a column with the strain. We have a column with the core RMSD. So when we add this group to that position, how much does it change the core RMSD? So you don't really want the RMSD to be changed too dramatically. So we can sort by RT-CNN score, and you see that again it finds halogens there as being good. It finds the alkyne group, which is in the crystal structure. And then it has some other groups that you could try that could improve the binding there. If you want to filter this table, you could plot RT. There's in all tables in ICM, there's this plot button here. So you single click and we can plot RT-CNN score against strain and maybe color by core RMSD. And go okay. It gives you some unusual. This has a very high strain, so you just delete that one and just to improve the plot a little bit. And this one you can delete. But here we can see with in this sector here, we can make a selection, and these are high scoring as well as very less strain molecules. So yes, that's the find group option. So you need to select an atom and then choose find group. So we go back to the original ligand again. So there's also the possibility that you may not like editing in 3D. And we found that when we're working with the chemists at Novartis, they actually prefer editing in 2D. So you can click on this button and it will open the 2D editor panel here. So you can delete groups. You see that the properties are calculated on the fly. I know that some of the inhibitors have a morpholine group at this point, so we can sketch that here. And then to send this to the 3D and dock it, you can just click on this button here, and that will place dock the ligand into the structure. So now you can see that we have the morpholine group here, and we have the score reported here. So we could add this one to our spreadsheet to save it as well. Just go here, and that will add it to this ligands table. So we have the morpholine group here. So you can also rotate torsions just by clicking on the rotate torsion button here, and just grabbing it and rotate. So you can see the ring is green in this position, but as we rotate it, it goes orange or smaller. That shows that the strain is increasing as you're rotating manually. But it's a good tool to maybe check out whether your group is actually can fit. Maybe there needs to be some movement of the side chains, or maybe it's just not in an optimal binding position. So yeah, so that's the torsion changes. These other buttons are for setting tethers and distance restraints when you're docking. So we're going to dock another EGFR ligand to this receptor. So to do that, we can you could read an SD file if you wanted to, SDF file, and choose the option dock table, because when you read it, it will dock it. I'll show that in a minute. But we can go to our 2D editor here and go to the chemical dictionary search button here and type in gefitinib and just click on it, and it's loaded the structure. So to do an initial docking and placement in the pocket, we can just click on this button, apply changes to 3D. And so now we'll see gefitinib in the pocket, but we need to really dock it. So to do a full docking. So we have an option here called redock ligand in current receptor, this button here, and then that would dock gefitinib to the pocket. So you can see that when we're docking, the ligand is fully flexible. And it's jumping around the pocket. It's a bias probability Monte Carlo method. And finally, it's docked. It's a little bit slower because I showed all the steps in the graphics. But generally, you wouldn't have that option checked, but it's kind of just for the webinar kind of thing. And you see that we have we have 5 conformations of the dock ligand ranked by score. You can overlay them by clicking in this box here. You want to see them. But it might be interesting to compare the docked ligand with the crystal structure of gefitinib bound to EGFR. So if we go to the search button here, and I know the code for the complex of gefitinib and is 2ITZ here. Click the binoculars button. And so that loaded the structure. So you can see in the workspace now we have our original PDB 1M17 with our inhibitor bound, and this one has gefitinib 2ITC bound. These two objects are for the ligand editor. So what we could do is we can superimpose the structure, the two structures. So if you double click and hold Control or Command and Mac, and we select both structures, we go to, you can see the selection here is green crosses. So we have the two structures, they're not superimposed. So we're going to superimpose this one onto this one. And you can see that in the ICM workspace, they're selected blue and they're green crosses here. We go to the superimpose button here. It asks us which one we want to be static. And we'll choose the original one we're working with, 1M17 as the static structure. Go OK. And so you see it's now superimposed. And we can compare the docked ligand. So the docked one is this one. Sorry, this one. And the crystal structure ligand is in magenta. So let me just so you can see it docks the core very well. The morpholine group, it prefers the docked one, sort of comes out here, probably because this crystal structure is a little bit different in this region compared to the one where gefitinib is bound. But overall it docks it reasonably pretty well. This obviously this group is pointing out into the solvent, so it can obviously have multiple different conformations to it. And if you look at the electron density, it's not particularly strong in this crystal structure in this region, but overall it docks the ligand pretty well. So this is the docked ligand. If we wanted to dock a small database of chemicals, of EGFR inhibitors, for example, to the pocket, we can do that by, just going to clean this up a little bit, the display. And made so many modifications. So if you go back to the Ligedit, we go to our history, modification history, and just go back to the original modification history table here, go back to our original ligand, alotinib, shown here. So if you wanted to dock a database of, we'll actually cover virtual screening and docking in future webinars. But I just wanted to show you that you can do it in the ligand editor. So you could go File, Open, and read in your SD file. And you can go to Molescreen and choose, or this option enables us to get all the known binders to EGFR from the Chembl database. So we get, it loads this table shown here. It says all the known or known targets from Chembl. We're interested in EGFR, so we can go here. Let's type EGFR, and we find EGFR human. We have 9,500 chemicals that are defined as binders to the EGFR ligand. We can just get those from Chembl. Shown here. And so this is ranked by PKD. So these are very, very strong binders. So what we're going to do in future webinars, we're going to do virtual screening and screen and score these compounds. But if you wanted to, we can cluster these quite quickly. And we get a tree. We can make a small selection here. Maybe the top four binders, or we can make selection in the tree on the right. But we'll also cover this in another webinar. But we're going new. This gives us these four. Then you can go to advanced, and you can choose the option dock table, and it will dock all these ligands to the pocket. And so you choose the table you want to dock. We've got quite a few different tables with chemicals. So if you have some ligands that you like that you put into your ligands table, you could redock them on the fly here. We can minimize only. The effort is the length of the docking simulation. We'll discuss that in 2 weeks' time. And you can dock them and you'll get a hit list shown here. But this is kind of moving away from what the topics are in this webinar. But we mentioned that one of the problems with this first generation of EGFR inhibitors was that this gatekeeper residue, this threonine 766, shown here, mutates, and generally, and it mutates to a methionine. So it's basically and we can see this graphically in the 3D ligand editor. If we mutate this residue, go to advanced, and then choose the option mutate amino acid, and we can choose from the residues here. We'll choose the methionine. When we do that, we can quite clearly see that it clashes. Oh, sorry, I mutated them wrong. Just 1 second. Let me just mutated the wrong residue. Just 1 second. I'll go back. So this is our this is the position we're at with it set up. So if we mutate the methionine here, sorry, the threonine. So let me just check the numbering. I can't remember the threonine 790, but I think it's different numbering in this. There's the gatekeeper residue, so it should be at the back here somewhere. Yeah, this one. So if we mutate that to methionine, you right click and choose mutate. We should see yeah. So we now see that because it's a methionine is significantly bigger than the threonine, it makes a clash with this alkyne group, and the makes the back hydrophobic pocket a lot smaller. So you get a clash, and so you can make, if you wanted to try and design ones to avoid that mutation, you could make some modifications in this region or something like that. But we'll see the second generation EGFR inhibitors in the next webinars in this series. So just wanted to finish to show you that primarily the ligand editor it has a number of tools for medicinal chemists, but under the hood of in the advanced section, there are some sort of more CAD orientated tools where you can screen replacement groups. So in ICM, just to show you, you can, so if you wanted to maybe explore different core regions, you can go to the editor button, click on the scissors, and click on the region that you want to. So the grayed out region is the region that we'll try and replace, and to use these arrow buttons, the scissors here, give you this arrow which toggles the display. So the grayed out bit will be the bit that will be replaced. So you go to advanced and you choose find linker option, and we have inside ICM a database of 47,000 different linkers that you could screen, and then you can filter this if you wanted to by you can filter by substructure or by different properties, or you could just screen the whole database of linkers, and then you get a similar hit list as before. There are also advanced options to match particular pharmacophores. If you want to keep certain pharmacophores, you can score it as how well it matches those. You can also do, if you're trying to escape a pattern or something, you can just allow ICM to screen a large number of substituents at various different points around the ligand, so multiple positions, and just let it run overnight, or and then you'll get some suggestions. It's called hetero multiple position R group scan, and you can choose which groups you want to screen to those position. There's actually videos on all of this on YouTube if you want to look into it. But we're coming to the end of the webinar, so I'd just like to thank you very